Welcome to Argo 2 Lesson 40. Talking about LDLs, several different kinds. There's something called chylomicrons. There's VLDLs, IDLs, LDLs, HDLs. So there's various different types of lipoproteins, which all carry non-soluble things around in the body. Right? So we've got something called a chylomicron, and that is going to carry triglycerides around in the body. How? Not always. Doesn't always carry triglycerides. You eat food, it goes into your stomach, and then stomach chyme. You have various kinds of enzymes like pepsin and things that break down proteins, and then that goes into your intestine, the small intestines. And then in the small intestines, bile salts are released, along with other things, pancreatic lipases, stuff like that. And they break down triglycerides, and they go into your mucosal cells, and in there they get reassembled. And to move around through your lymphatic system and then in your blood system, you need something to carry this thing, because this thing is insoluble in aqueous media, so it's just going to go and get stuck in plasma membranes. So you have basically what is the equivalent of a micelle, but isn't exactly soap, because I think I've mentioned in the previous lesson that something like soap would be insanely dangerous, denatured proteins, right? So, but in the same kind of an idea where you have some sort of like a particle that's made up of stuff that inside is hydrophobic and outside is hydrophilic, and it would be used to carry around things like this. So a chylomicron carries around triglycerides, but also has cholesterol, also has a lot of other things in it. And then there's all the other ones that have names that don't sound like this. And they're called lipoproteins. And a chylomicron is a lipoprotein also. It's just this one is the lowest density, and then the density increases to low density, intermediate density, up to high density lipoproteins. So all the other ones have some kind of lipoprotein name, and chylomicron doesn't sound like that at all, it's just chylomicron. This thing moves around triglycerides, and those are really for like cholesterol types of deliveries, either move cholesterol to cells or take cholesterol back from cells. And I think that's about it. Like, to go any deeper than that kind of doesn't make sense in this course. Did anybody do the simple assignment, like look at the prostaglandin and figure out chiral centers, R and S? Some simple stuff that you can do. So what else is there? We didn't really talk about the fat-soluble vitamins, but you could read about those on your own. And we mentioned about retinal, for example, which actually is not a vitamin. Retinol is the vitamin. Retinal is a derivative of the vitamin, vitamin A. And you could take a look at their structure, see what things are similar with their structure. For example, for some reason, the fat-soluble vitamins, besides vitamin D, have 4 isoprene units. There seems to be like this magic 4 isoprene unit number. Okay. So that moves us into a totally different chapter, which is amino acids. I was going to just jump right into polymers, but I figured I have to talk about amino acids first, because there's some ideas within this chapter that we need before we talk about polymers. When talking about polymers, we want to think about how the macro structure, you take a look at this marker, for example, it's made out of some plastic, how that, what that has to do with the microscopic subunits of the polymer. So we want to know about the individual monomers, and we want to know how those monomers link when we talk about polymers. So we're thinking about the linkage between them, but that's not really where all the strength in a polymer comes in. The strength in a polymer sort of comes in with how these chains interact with each other. So there's like two things to notice, the connection between them and the connection between chains. And that governs so some of the connections we're not there yet. Like, we don't know what a carbamate bond is, so we need to think about that in this chapter. So let's begin with amino acids. You cool with that? But one thing that's good about what we've done in the past is that we know the Henderson-Hasselbach equation, I hope. Anyone heard of the Henderson-Hasselbach equation? Okay. We know the Henderson-Hasselbach equation, so we are not lost in terms of that. And we could then understand like the structure of a particular amino acid at physiological pH. So there are 20 naturally occurring amino acids. There's way more than that. There's way more than 20 amino acids, but there's 20. Why we talk about 20? There's 20 naturally occurring amino acids that participate in peptide bonding to make proteins. So, like, of course, you could find proteins with some of the ones that are not these 20, but generally there's 20 amino acids that proteins are built out of. And these amino acids, 19 of them are L-amino acids for the naturally occurring stuff. So sugars tend to be D sugars. The more common sugars in biology are D sugars. And the more common amino acids are L-amino acids. What does L-amino acids mean? They rotate light to the left. No, that's absolutely not what it means. That they have the S chiral center. No, that's not what that means either. That they are derived from L-glyceraldehyde. Yes, that's what that means. And if they had no connection with glyceraldehyde at all, then we wouldn't call them L anything or D anything. It's just the connection with glyceraldehyde. So what does the L-amino acid mean to us? We draw amino acids from the N-terminal to the C-terminal. N on the left side, C on the right side, and that's, so for some reason, that's not the way that amino acids are tabulated. Like you look at a table of amino acids and they're not at all drawn that way on a lot of tables. But then you look at peptide structure later in our textbook in the chapter and many other textbooks also. What I'm saying is they have like some weird, they'll show a side chain and then they'll show like a COO minus pointing up and an N pointing this way. That's not at all from the what is the N-terminal? So the amino acids are alpha amino acids. Besides being L-amino acids, they're alpha amino acids. And what that means is there is a amine group that is attached to a carbon. And if it's attached to a carbon, that first carbon's the alpha carbon, right? Alpha, beta, gamma. On that same alpha carbon is another functional group, COO minus. So the COO minus, the carboxylic acid, is alpha to the amine, or the amine is alpha to the carboxylic acid. And this is the N-terminal, the nitrogen end of things. And this is the C-terminal, the carboxylate end of things. And we draw them left to right, because in this country, we're writing English, right? We write left to right. If there's some other place where they write right to left, I would imagine they would have the N-terminal on the right side and the C-terminal on the left side. I'm not saying it's illegal to draw it the other way. Don't get me wrong. You could write OOC, you could do this. That's no issue. But we're going to see there's like a particular structure that we have for L-amino acids where we start by drawing it that way. Why do we care about the N-terminal to the left and the C-terminal to the right? In the next chapter that we cover after this, I don't know if we'll get to it. We'll see how far we get today. So we might skip DNA, RNA. We talked a little bit about this already, and we'll just maybe throw in gel electrophoresis. Like that part we'll definitely talk about. But when it comes to amino acids or proteins, we draw them from the N-terminal to the C-terminal. And when it comes to writing nucleic acid sequences, like RNA or DNA, we always write them from a 5' to a 3' direction. So what I'm saying is, if I was going to write like some little code in RNA, A, U, C, C, G, A, C, U, A, right? Let's say there was that code. They are linked right? RNA nucleotides are linked by phosphodiester linkages from the 3 to 5 prime. So here is a ribose, which this is carbon 1, 2, 3, 4, 5, right? And over here would be some nitrogenous base. This is not the first time I drew this. And over here is an OH and an OH. There's ribose. And then here, there would be a carbon with, let's say, an OH. So this is going to be a nucleoside, and I put a phosphate on there. It's called a nucleotide, the monomer unit of the polymer called the nucleic acid, RNA. And it would be linked in a phosphodiester bond, so this would be part of a phosphoester, and this would be connected to an oxygen of the next 3 prime. Do you see it'll just keep going in that direction? Could you picture that? Right? Well, we've already said a while ago that DNA and RNA are synthesized from the 5 to the 3 prime direction. So because they're synthesized in the 5 to 3 prime direction, this is how we draw it on paper. So if I write AUCCGACUA, and I don't tell you which side is which, that means that we all assume this is 5 prime and this is 3 prime. Now, if I do label it, I could label it, and that's fine. I could call this the 3 prime. That's allowed. But unlabeled, that's the 5 prime, and that's the 3 prime. So if I draw a peptide, if I draw, let's say, a tripeptide, let's say I draw histidine, isoleucine, serine, cysteine, threonine, and I don't tell you which side is which, that means over here is the N terminal, and over here is the C terminal. I could label it otherwise. That's possible, but without no. And why? Because this is how peptides are synthesized. The ribosome connects things starting from the nitrogen end towards the carboxylate end, doing a dehydration reaction in the process. So for that reason, I'm going to draw the amino acids that way also. I'm going to put an H3N, and then you draw a corner. Guys with me? I could draw the corner up, which I will do, and then write COO minus, or I could draw the corner down and write COO minus. Fine. Now what differs in these 19, or well, actually all 20 amino acids, is what goes on over here. The R group that goes on to the alpha carbon. 1 out of 20 is called glycine and looks just like this. It has a hydrogen and a hydrogen, 2H. So that's the simplest amino acid called glycine. That would be strange to call it L-glycine because D-glycine would be the same exact thing. It has no stereochem at all. So it's just glycine. But then the other 19 amino acids, they have an R group over here. Now this is what makes them L-amino acids. When drawn in this way, from the N on this side, C on that side, corner points up, the R group comes out of the board. That is going to be the same in all 19 L-amino acids. Do you hear what I'm saying? So they're not always going to be R or S or anything like that, but the spatial direction of these groups is going to be the same. That's what makes them the 19 L-amino acids. So if it's like that, if I drew the corner down, then where's the group? Is it still out of the board? This is some simple stuff. Like you're done with chapter 3, right? We passed chapter 3. Yes, of course. Flip it over, it goes into the board. So let's say I draw it the other way. Let's say I go H3N on this side, and then I go corner up, and I go COO minus this way. Where's the R group? Out of the board, into the board. That's like taking that one and turning it around. The R group is into the board. Simple enough. There's one more combination. So just to exemplify the fact that L does not mean R or S, it just means that it's derived from L-glyceraldehyde. Here are 2 amino acids that are very similar to one another. This one is called L-Serine. It is one of the amino acids, which is the reason that high glucose concentrations in blood might be a problem. Why is it one of the amino acids? Because when this is part of a protein, then this nitrogen is part of a peptide bond, an amide bond, and this COO is not a COO because it's part of an amide bond. So there's like a chain going, and this is the side chain that comes off, and this is an alcohol side chain. So you realize if this is part of a protein and over here was this amino acid, then you would have an OH sticking out. And if you have large glucose concentration in blood, then of course that hemiacetal could react with that and make an acetal. By the way, did anyone get the question of the day? Did anyone see this question before? Raise your hand. Just made you raise your hand for no reason. I didn't tell you why to raise your hand. Okay, raise your hand if you've seen this question before. Okay, did you answer it? It's not that difficult. Just realize what you're looking at, and not. It could be impossible. Let me take that back. It's either not that difficult or it's completely impossible. You're either doing some organic chemistry or you're flipping through your book asking yourself where I've seen this before. What do I have in front of me? I have a hemiacetal. What the hell does that react with? Alcohols. What else? I have alcohols. What do I have here? An aldehyde. In this relationship, the aldehyde's the electrophile. So I need some potential nukes, let's say. How about alcohols? Now over here, there's a whole bunch of diols, let's say. So if I had an aldehyde that reacted with one alcohol, it'd make a hemiacetal. If I reacted with a diol, I could get an acetal. Do you follow? If this OH is equatorial or axial, that OH and this OH are vicinal. So if they both link to that carbon, you're going to get a 1, 2, 3, 4, 5-membered ring. If this OH and this OH link to that carbon, you'll get a 1, 2, 3, 4, 5-membered ring. You keep doing this until you realize when these two OH link to that carbon, you get a 1, 2, 3, 4, 5, 6-membered ring. And that's where the reaction occurs. Do you see it? What I drew there? Oh, when it links, that aldehyde carbon can be R or S, right? Everyone see that? So this will be either axial or kind of equatorial. Okay. Now that I've distracted you, back to this. So this is L-Serine. And then there's an amino acid that's super close to this one. CH2SH. Why did I call that super close? Because it's really one atom replacement. It's not even like a super different functional group. It's a one atom difference. And the atom is still a chalcogen. It's still in the same column. Only difference is this can be a hydrogen bond donor. This cannot be a hydrogen bond donor. In some cases, that's going to be a big difference. This is not acidic. I mean, everything's acidic, but this is not acidic within a 0 to 14 pH. And this is a pK of around 8. 8? You know the reference I'm talking about? Okay. L-Cysteine. L-Cysteine. Did I spell that correctly? Think there's no E? Now I know there is an E. And I did spell this correctly. But is that right? Do I have to put the I first, the E first? Where does it go? What was the rule? I before E, except after C. Well, there's a C. No, that's not how it goes, because it's supposed to be right after C. I before E, except after C. Who knows that there's more words to that? If this was 6 years ago and I said I before E, except after C, half of the room would finish the sentence. How many of you have heard the I before E at all, any part of that? Only 3 people in the room have heard the saying I before E. It used to be 100% of the room heard I before E and 50% of them knew how to finish it. Now 3 people have heard I before E and don't even know that it finishes. I before E except, yes, I'm going to teach it. I before E except after C, or when... I could stop the lesson now and they just don't teach anything else anymore, right? So maybe we want to learn what I have to teach. Hold on, the fire trucks need their time. It's I before E except after C, or when sounding like A, as in hey or away. Who's ever heard of that before? Usually when you have I-E words, it's I before E, right? I before E, except after C. So if there's ever a C followed by, then it's an EI. I before E, except after C. But that's not it. There's more. Or when sounding like the letter A, as in hey or away. Now, that always bothered me, because why use hey or away when they don't have an EI in them? We could use the word way. Or something like freight, where you have an EI, right? Now, once we know that full saying, I'm going to add to it my version. I before E except after C, or when sounding like A, as in hey or away, or in bio, like in protein or cysteine or other ene. Got it? You're good? So this is correctly spelled cysteine. There's a different thing called cysteine, and that's when you have 2 of these with a disulfide bond between them, and cysteine is missing the E. But cysteine has the E. That's L-Serine. This is L-Cysteine. They're both L-amino acids. They're both structurally similar in spatial orientation. However, they don't have the same R and S. Here's a chiral center. Here's a chiral center. And if you find the R and S at the chiral center, there's an H. There's an H. Top priority is what? N, H, C, or C? N. So this is number 1. Then it's C versus C. That's the same. You know what's frightening is that people at this level I run into don't understand how to do priority. It happens every semester. As far as I could go back in teaching, people reach this level and still can't figure out priority. Carbon versus carbon, that's the same. So what do? You add up the mass. No, that's not it. You go atom by atom until there's a point of difference. Now, there is a point of difference between carbon and carbon. This one has 2 H's and an O, and this one has 3 O's. You cancel an O with an O. You go to the next highest priority. This has more O. This has only H. This is a greater priority. This is 2. This is 3. It goes 1 to 2 to 3. Counterclockwise, lowest is in the back. This is S. L-Cysteine, this is 1. Carbon versus carbon, that's the same. Next highest priority, this has hydrogen, hydrogen, and sulfur. This has oxygen, oxygen, and oxygen. Sulfur is a greater priority than any oxygen. Therefore, this is number 2. This is number 3. This is R. L-Serine is S. L-Cysteine is R. Dare I say, memorize that for anyone who doesn't know how to figure out priority. Do it if you need to do it. If you're my student, you're going to do the following. You are going to remember the 20 amino acids, all of them, all of their frickin' structures. I know that's not required now, but let me give you like a history of like what happened, right? There was a time where these chapters were required, start to finish. There was a time, I'm not saying like 60,000 years ago, I'm talking about a couple of years ago, where in the college that the people in this room currently go to, these chapters in depth were required. Strange how like time passed, you remove more stuff and it's more difficult on the students. I don't get it. But anyway, there was a time where this was required, and my students learned every one of the amino acids, every single one of them, all their structures, all their pK, every single one of them. Then they took Biochem 1 and they realized they forgot a lot of it, most of it. They learned it again in Biochem 1. Then comes Biochem 2, they realized there's a couple of them they still didn't remember. They remembered it again. They get to med school and they just fluent in it, absolute fluency. What happens now? I ask my students to remember these. 1 or 2 people care to do it. The rest don't. They get to medical school, they can't even draw this. They can't even draw nitrogen, alpha carbon, carboxylic acid, and they wonder why they're flunking out. So anyway, if you're my student, if you're a real student of mine, you're going to learn the 20 amino acids this semester and allow yourself to forget them. That's totally fine. So we can learn them again next semester, and that's how it's supposed to go. So that there's a point later where people around you are struggling, and you're like, it's not so hard to learn this. Well, it's because you learned it once before, and now it's like this muscle memory that's coming back to you. Now, of course, where do you begin? It's maybe a little daunting to just randomly memorize structure. So there is a really good place to begin. There's a lot of amino acids that have side chains that are just like neutral. For example, you have alanine, which is a methyl group. You have leucine, which is a couple of carbons. You have isoleucine, which is an isomer of valine, branched. It's like same kind of hydrophobic side chains, right? We have 7 amino acids that have ionizable side chains. And those ionizable side chains, they can help function in proteins. They can do things. They can do your nucleophilic substitutions, eliminations, acid-base reactions. So what we want to do is study those 7 first. We want to begin with those 7, the ones that have ionizable side chains. And once we know those well, then we could start adding to our database. Right? We want to know the structures of all 20 amino acids. So we want to know all of them. We want to know their structure. We want to know the pKa for all of them. The pKa values. Now what the hell does that mean? It's a weird thing to say. So if you're somebody who already knows, if you're watching this video and you're somebody who already knows something about amino acids, what do you mean the pKa of them? There's many pK values, I know. So every amino acid, when fully protonated, has a pKa for its alpha carboxylic acid group, and it's got a pK for its alpha amino group. And besides that, 7 of them have pK for ionizable side chains. So that's 47 pKa, right? There's 20 amino acids, 20 alpha carboxylic acid groups, 20 alpha amino groups, and 47 pK. You're going to really remember 47? I don't need you to remember every single one of them. What we could do is just say, there's a carboxylic acid group that has a pK between 4 and 5, right? Well, not this one, because this has a close inductive effect. This is what makes sense between 2 and 3. We've talked about that back in Orgo 1, where we showed butanoic acid, and I said if this has a pK of 4.8, and if I put a Cl over here, it's like 4.6. And if I put a Cl over here, then it's like 3.8. And if I put a Cl over here, then it's like 2.8. Remember 2 and 3, the closest you could get? So, with that being said, this carboxylic acid, alpha carboxylic acid group, has a pK between 2 and 3. Now there's a table of pK in the beginning of this chapter, and you could look at all the pK of every single alpha carboxylic acid group, and some of them are not between 2 and 3. Some of them are slightly like outliers. So what we want to know is which have pKa values that are less than 2, which ones, and most important, homework assignment is why do they have those pK values? Any idea what the hell I'm talking about? Which amino acids have an alpha COOH pKa less than 2, and why? Do they have that? And again, here's a moment where we're going to start abandoning organic chemistry and thinking it's just like some kind of like supernatural phenomenon. No, it's like if I show histidine, I'll give you away one of the secrets here. Here is this amino acid, histidine, fully protonated. It has 3 pKa. This one is less than 2. It's 1.8. Then the side chain is a 6.0. And then the alpha amino group is approximately a 9. Why the hell is that less than 2? So let's think about it. Do you notice that when it's fully protonated, when I drop it into very strong acid, this is protonated, that nitrogen is protonated, that nitrogen is protonated. Do you notice it's got a positive charge and a positive charge in very close proximity to one another? This terribly repulsive positive charge. So wouldn't it be very beneficial if you could just quickly deprotonate that and drop a negative in between to sort of like break up the positive? How the negative attracts that, right? Do you see why the pKa would be? Remember that an acid is acidic depending on the stability of the conjugate base. And the more stable the conjugate base, the more acidic, the more it wants to become that conjugate base. This is not going to be so stable with a neutral over here. It'll be more stable with a negative over there. Do you see? When I'm saying why, I'm asking you to like come up with some random bullshit. I'm asking you to think about chapter 4, right? And think size, resonance, inductive effect. What have we added this semester to that 5 effects? 2 things. Aromaticity and solvent interactions, right? So there's that. Then on this side, you've got an alpha amine group. Now amines, this is a primary amine. An alpha amine should be something like 9 to 10. And it is. This is between 9 and 10. And you could look down the list of all of the amino acids that have... here's what I want you to do, though. You should use the organic chemistry textbook for this. I've given an assignment like this before, and some students have like the biochem book, and they're using that. You'll notice that the numbers are different in the biochem book than they are in the organic chemistry book. How could they be different? Because there's different labs measuring this. These are measured numbers. You can't... There's no like... It's not real universal numbers. They have to be measured according, right? It wasn't like Moses came down with 11 commandments. The 11th one was the pKa values of amino acids. That would be great, because then there would be no discrepancy. Every book would have the same numbers, but they don't have the same numbers. Right, so with that being said, use the organic chemistry book for this purpose. I want to know which ones are unusually acidic and why. What am I going to think about on this side? There could be some that fall out of the range and go below 9, and some that fall out of the range and go above 10. Which one am I caring about? That's an acid, so I want to know which is unusually acidic and why. This is a base. I want to know which one is unusually basic and why. So I'm looking what goes above 10 and why. There's not going to be a lot of them. It's going to be like 2 or 3 and 2 or 3. But you figuring out why is you really kind of digging in and applying the orgo to the biochem. Do you hear what I'm saying? And then we have the side chains. Okay, so what do we need to know for somebody who's going to really study this? All the pKas, you're not going to really know 20. You're just going to know they're between 2 and 3. I'll call it 2.5, except for those couple that are beneath 2. I'll call this 9.5, except for those few that are above 10. I want to know the name, the full name of each amino acid. Right? For example, the one I drew over here was serine. Then there's a three-letter code. The three-letter code is really easy because it's almost always the first three letters. So it's SER. And then there's a one-letter code, and it's not always so easy. For serine, it is S. And for alanine, it's A. But for tyrosine, it's Y. And why is tyrosine Y? Because there's threonine, there's tryptophan, there's a lot of other T letters. So because of that, so we want to know the three-letter code, the one-letter code. I'm not kidding. Like the last thing you need while you're doing complex ideas, thinking complex pathways, is to be the one in the room who needs to memorize the amino acids. While everybody else is fluent and knows and is thinking deeply through pathways, you're trying to figure out the name of structure. It's like being the person in Chem 1 who's trying to figure out the algebra. You can't win that way. So one thing that you could do to remember the single letter code, right, is to send each other messages. Right? So you text your friend in three-letter code, and they have to decipher it in one-letter code, right? So you write serine, histidine, isoleucine, threonine. S-H-I-T. Right? Stuff like that. And then you'll remember you can't make every word because we're missing some letters. There's only 20 amino acids. And there's 29 letters of the English alphabet. It's funny how some of you are actually thinking about this right now. Anyway. Everyone knows it's 35. Okay, moving on. Wow. I guess orgo's not our problem. There's not 29. That was not that. Holy crap. Okay, what we need to do is not believe every word out of somebody's mouth, because there's something called a typo that happens, and then you'll be stuck memorizing a typo for 55 years, right? I could have tried to write 2 to 3 and wrote 0.2. That's it. Now it's 0.2. That's not how it works. We can't be so nervous that you can't miss a single thing the person says. Okay, we want to think about the side chains, the ionizable side chains. And there are 7. So the 7 ionizable amino acids. I'll create a little table. Let's do this. Let's have the name. Then after that, we're going to have the side chain. Then after that, we're going to have the pKa. And then the charge of the side chain when protonated. I was going to make another column of charge of side chain when deprotonated, but that's a little ridiculous. Because just remove a proton, right? Like if I said the charge of the side chain when protonated is +6, then deprotonated would be +5, right? I should have just said +20 after that and watch some of us collapse, right? There's 39 letters of the English. Okay. I'm going to go in order of lowest pKa to the highest pKa. And so the first amino acid is called aspartic acid. Aspartic acid. You know, not all amino acids have the name acid in them, right? So like there's amino acids called serine and histidine and tyrosine. This one is called aspartic acid. So that kind of helps. It's an acidic amino acid. Aspartic acid has a 3-letter code, ASP, and a single-letter code, D, of all things. The person who made up the single-letter code, they said that that person was from Brooklyn and had a Brooklyn accent, so the reason they chose D for aspartic acid was because they pronounce it aspartic acid with a D. I don't think that's true at all. Well, that's what you might read on a website. What happened? There's a whole thing. There's a whole field called bioinformatics where back in like the 1960s and so on, we had computers, but we didn't have flash drives like we have right now. What we had was punch cards. Anybody ever heard of this? That you had these cards that each letter, like if you're typing something in, each letter was a card with certain holes in it, and you put that into the computer would read it, you put the next card in, read it. So if you had a protein that you wanted to keep the information for a protein, you'd have to have all of the cards, stacks of cards, that were all the names. So that's why there was a 3-letter code in the first place, that instead of using the whole name aspartic acid, they would use ASP. You'd only need 3 cards, right? So you'd use the card A, then S, then P, then the next amino acid, the 3 cards, then the next amino acid, the 3 cards. You'd store that information in the computer. It turns out that like after a while, we got a lot of proteins in the databases. We'd have warehouses with like floor-to-ceiling cards stacked. So a famous biochemist was up one night. It was like 2 o'clock in the morning. And she realized we're very low on space in the warehouse. There's like, oh, the rooms are filled floor to ceiling. Why are we using 3-letter code for these amino acids when we have more than enough letters in the English alphabet to assign 1 letter per 20 amino acids? So she's up in the middle of the night and she's trying to figure out which amino acid has what. So alanine got the letter A. So aspartic acid couldn't be A. And S was already taken for serine, and P was taken for proline, and A was, again, alanine, and R was taken for arginine, and arginine doesn't start with the letter R. Arginine starts with the letter A, but A was taken for alanine. So she had... I think the reason she chose D for aspartic acid was because there's 2 acidic amino acids. One of them is called aspartic acid, and the other one's called glutamic acid. And those 2 acidic amino acids, I think she wanted to just put them together. And the letters, the only consecutive letters that were available that were not used for anything else was the letters D and E. So she chose those consecutive letters, glutamic acid, which is GLU, and the letter E, D and E. I think that's why she chose it. Never told anybody. There's no like actual official account of, not to my knowledge. So what is aspartic acid? I guess I'm going to draw the entire amino acid. That's an H3N plus alpha carbon, COO minus, and then this side chain is 1, and then a second carbon. The second carbon is COO minus. So you know what? Another way to call aspartic acid, if we're going to do orgo, we're going to name it orgo-wise, you would call it alpha amino succinic acid, or alpha amino succinate. Do you see if I cover this up? Isn't this succinate? Oh my such, 2 carbons in between? And on one of the neighboring carbons is an amino group, right? So alpha amino succinate. Glutamic acid is H3N plus COO minus, and literally the same thing except 1 extra carbon. 1, 2, and then a COO minus. So it's 3 carbons in between carboxylic. COO minus with an H3N plus. By the way, I didn't draw this in the way I'm supposed to. I didn't draw this N terminal to C terminal. I didn't do that. I drew this just to like link on the nitrogen from orgo, right? I'm not doing biochem. So that's like a succinate with an alpha amine, and if it's glutamic, then it's going to be glutarate. See extra carbon? Oh my, such good. With H3N plus, right? We with that? There's a really important reason why I'm giving you this table. We'll see shortly. So when protonated, what is that side chain? Maybe I should have done that. I'm drawing these like at physiological pH. Let's change this to fully protonated. And we're just looking at the side chain. We're not looking at this or that, right? The side chain pKa is 3.9 for aspartic acid. And the side chain pK for glutamic acid is between 4 and 4.1. I'm just going to write 4.1. That makes sense, right? It's a little bit further away, weaker inductive effect. Right? Look, the nitrogen is withdrawing from that, right? But here it's withdrawing less. So there's aspartic, there's glutamic. And then after glutamic, there's histidine. Single letter, a three-letter code is his, single letter code H. And you know what? Because there's not a lot of room in here, I'm going to draw it right here. So what does histidine look like? It's H3N plus, corner up, COOH. I'm fully protonating it. COOH. And then, I don't want to draw this on a wedge, so I'm going to do something else. I'm going to draw it without stereochem, and then I'm going to draw the H into the board, and that's the same thing as putting it on a wedge, right? It goes carbon, and then after the carbon is a 5-membered ring. That 5-membered ring has 2 nitrogen in it. So watch how I draw it. This carbon is going to be part of the 5-membered ring. To one direction, draw carbon. To the other direction, draw nitrogen. Right? After the carbon and the nitrogen is a carbon, and they connect at a nitrogen. No, something is wrong. I take that back. I wasn't supposed to say that. Because I made a 6-membered ring. It's a 5-membered ring. One way is a nitrogen, the other way is a carbon. What I was supposed to say is after the nitrogen's a carbon, after the carbon's a nitrogen. So after that is a carbon, after that is a nitrogen, and then they link up together. Nitrogen, carbon, and then after that carbon, nitrogen, right? And now this thing needs to be aromatic, because this has an aromatic, and it's interesting because this isn't counted as one of the aromatic. If you look at a table of amino acids, there's the aromatic amino acids. And this one is not considered the aromatic amino acid, even though it is. We looked at this Emitter Salt ring before. We said which nitrogen is more basic and all of that, right? We drew it sideways with the p orbitals. Why is it not considered aromatic? Because it's considered a basic amino acid. Like overrides its aromatic nature. The way to figure out where to put the bond, which is, it's really pointless what I'm showing you here because you just make it aromatic, protonate it, because since there's going to be resonance. But the standard way of doing it is put a double bond between the carbons. The carbon to carbon, make a double bond. So over here, make a double bond, and then conjugate it. So over here, I'm going to put that, right? And that means this nitrogen has a hydrogen on it and a lone pair that's part of the aromatic sextet. And now this other nitrogen is going to be connected to an H and have a positive formal charge. Histidine is the only amino acid that is like really confusing on the actual table because all of the amino acids on the table are drawn at physiological pH. So what that means is they're drawn at pH 7. So any group that has a pKa less than 7 is going to be deprotonated. Based on the Henderson-Hasselbach equation, when the pH equals the pK, it's 50-50 protonated, deprotonated. And when the pH is above that, more of it is deprotonated. So any group that is more acidic than a pH of 7 is going to be deprotonated, and any group that has a pKa greater than 7 is going to stay protonated. So it's every other one is going to make sense for students except this one because it's shown like this. So when you have the other basic amino acids, they have a positive charge in that nitrogen ring or chain or whatever. For example, there's lysine, there's arginine, they're going to have a positive charge in there. But this one has a neutral side chain. It's always confused students where to put the other nitrogen when protonating. So if this is what you're starting with and I put acid into this flask, don't protonate that nitrogen. We've talked about this in the past, right? You protonate this one. That has the actual electrons orthogonal right? And then you would protonate this. So make sure you know how to draw it fully protonated so that it doesn't confuse you. It's going to confuse you at some point whenever you sit down and really try to process this, is what I'm saying. So this has got a name for the side chain. It's actually called, this thing right here is called an imidazole. And the pKa of the side chain is about 6, approximately 6.0. Oh, and if we want to make it an L-amino acid, before I erased it, I should have put an H back here, right? So we have that, and then we have cysteine. 3-letter code CYS, single letter code C. And the side chain is a sulfhydryl. Sulfhydryl. Which we've already drawn cysteine on the board before. We figured out that it was R rather than S, right? It's just one carbon and then an SH. That's all that is. And that has a pKa of 8.0. I'm a biochemist. Like that's where my degree is. That's what I've done years and years of research in. So can you imagine that, like, you know, besides everything else that I've demonstrated that I understand the know, right, for you. Can you imagine how many times I've stared at a frickin' cysteine with side chain 8? So then you get an Orgo 1 student saying, my professor said it's 10 or 20 or something. It's just like, holy crap. Anyway. It's tough. What comes after cysteine? There's 3 more, right? We've got 4 already. So there is, depending on which book you go with now, I'm going to go with our book. There is tyrosine. And tyrosine has a side chain, has a 3-letter code TYR, single letter code Y. Why? Because the T was taken. You know, the only one that has really a bizarre name is tryptophan. Not name, but a single letter code. The 3-letter code is TRY, right? But the single letter code is W. And that has like nothing to do with nothing, but we actually know why she picked W. All the letters are taken. T for threonine, R for arginine, Y for tyrosine, P for proline. Everything is taken, right? Except O. Should have used O. But anyway, so she was staring at this. It was late at night, and tryptophan has a side chain that looks like this. So this is serotonin. I mean, you know that an artist drew this and not any kind of chemist. They put an N and an H inside a ring. Like, where the hell are those? That doesn't matter. Serotonin comes from tryptophan. Dopamine, norepinephrine comes from tyrosine. So these amino acids are not just to link up and you know, make proteins. They also give you various kind of molecules. So she's staring at this, and she realizes it's the only amino acid that has 2 rings. So because it has 2 rings, she's thinking of the word 2. And T was already taken, so she chose W as the single letter code for tryptophan. Listen, she what was the major thing that she accomplished? The next day, instead of 3 punch cards for every amino acid, you need 1 punch card for every amino acid. Instead of 3 rooms being taken up in the warehouse, you have 1 room taken up in the warehouse. It was huge. So tryptophan is a W. Tyrosine, what is its side chain? It's actually a phenol. Because what it is, okay, so let's build tyrosine. This is a amino acid, just a regular amino acid, and if over here I put a methyl group, then that would be called alanine. It's the second simplest amino acid, because the simplest is glycine, just 2H. Alanine has a methyl. If on that alanine I put a benzene ring, it is called phenylalanine. You have used phenylalanine in Orgo 1, right? In lab, phenylalanine. So tyrosine is a phenylalanine with a OH, with a phenol. Tyrosine has an OH over there. Do you see that? We can't make tyrosine or phenylalanine. These are essential amino acids. We're going to talk about that one day. Over history, we've lost the ability to create certain amino acids. But if we have phenylalanine, we have the enzyme to take phenylalanine and transform it into tyrosine. But even still, we say tyrosine is an essential amino acid because we still can't make out phenylalanine. We need to ingest phenylalanine. What do you think you call the enzyme that is going to put an OH right here? It's called a cytochrome P450. Anything that replaces an H with an OH anywhere in the body is a cytochrome P450 kind of enzyme. It's very important for you to kind of embed that into your brain right now. What is the Sidechain Pka? As expected, 10.0. Do you notice that, like, why do we need 7 side chains? I would say we don't need 7, we kind of needed 6. Because 4 and 4 are very similar to each other. But can you see how amazing that the body can make a gradient? Look, if proteins are made up of amino acids, and amino acids are going to be the things that catalyze reactions, and a lot of the reactions we studied are acid or base catalyzed, don't you want the ability to have a protonate or deprotonate at every potential pH interval? So this is, and plus, you would say, but you don't have a 5. Well, inductive effects can make a 4 turn into a 5 or a 6 turn into a 5 based on the environment that they're in. So that works also. Just had a terrible thought. Okay, good. So the last 2 is lysine, which has a 3-letter code, LYS, and a single letter code, K. Why K? Because leucine took the letter L, so she chose the closest letter to an L for lysine. And M was already methionine, and N was asparagine, so she went the other way, and the letter before L was K. What is lysine side chain? I'm just going to draw lysine H3N plus. H3N plus, alpha carbon COOH. And from the alpha carbon, it's 1, 2, 3, 4 carbons, and then an NH3 plus. So from the alpha carbon, it's 1, 2, 3, 4 carbons, and an NH3 plus. So it's a primary amine. It's a primary amine. And what is the pKa of a primary amine? You should guess that it's between 9 and 11. So 10.5 makes sense. Now this is lysine. The last one is arginine. ARG, single letter code R. What's a pirate's favorite letter? R. I actually said that once, and students like, no, it isn't. It's C. I was like, that's unique. You know, like the C. C. Arginine and lysine are structurally similar. That's why certain proteins, digestive proteins like trypsin, mistake lysine for, well, not mistake, but recognize both lysine and arginine and cleave after them. We'll talk about digestive proteins and proteases. So they both have 4 carbons. It's just that arginine has one of the carbons embedded in this group called the guanidinium group, guanidine, which we have mentioned before. And arginine looks like this, H3N plus, alpha carbon COOH, and then there's 1, 2, 3 carbons, not 4, 3 carbons, and then after that, the guanidine group, which has a carbon in it. And the guanidine group goes like this. It's N, double bond, with an NH2 and an NH2, and that N has an H on it and a positive charge. There's more than one way to draw this. Because notice, this has a lone pair that could go this way and resonate that way, and that's a positive nitrogen, or this could resonate that way. Do you see what I'm showing you? That this has a lone pair that could resonate, put the positive charge here, or this could resonate, put the positive charge here. If that wasn't true, this would be so wrong for me to write 12.5 as the pKa. Why would that be wrong? Because a sp2 nitrogen is supposed to have a pK between 4 and 5. Not 12.5. So why the hell is it 12.5? Because resonance stabilizes this. And it's what? An acid. So if you stabilize an acid, you weaken the acid. pKa goes up. So this is arginine. What happens if you do this? What happens if water comes in here and cleaves right here? I mean, it happens in the body. It happens in your liver. It's happening right now. Anyone need to go to the bathroom? If water cleaves over here, you get an amino acid. First of all, you get this remnant. You get this amino acid that is not one of the 20 at all, but it's something that is in our body a lot. And it's called ornithine. Now what's the other piece? The other piece is H2N carbonyl NH2. Anybody ever seen this molecule before? No idea? This is called urea. And in our body, there's a lot of nitrogen waste because of proteins and nucleic acids that we eat. We have nitrogen cycles, we have nitrogen waste, and there is no way that this can be in our body. That's a horrific poison, because in the cell, as I mentioned before, everything has to be not reactive. You've got ATP, and I'm just going to write A, with O, P, O, P, O, P, O. What are these bonds that connect ATP together? They're called phosphor what? They're not diester. They're totally not diester. A carbonyl, oxygen carbonyl is not a diester. These are phosphor anhydride bonds. This is the top of the list of reactivity. And what happens if we have water? Shouldn't water come in and cleave an anhydride by itself? Is anybody with me here? It's tough teaching this when I see the look on people's faces thinking, I know he wants us to know stuff, and I really want to learn, but I have finals coming out. Like, that's the look I'm looking around the room. I'm seeing that look on people's faces. And I can't wait till your oh shit moment when you get the final exam and it's this. When have I been wrong in the frickin' past? Wake the hell up and learn what I'm teaching you. I've mentioned this 3 times in the past few lessons. That's an anhydride, and in chapter 18, water can cleave that, no problem. You don't need a catalyst. That would be, so that's the reason we don't have a regular anhydride in our cell. Because you can't have water just chop it up uncontrollably and have the cell explode. So instead of that, you replace the carbon here with a phosphorus, so it's O minus, O minus. Now that negative charge would never allow water to come near there until an enzyme protonates the carbonyl, so to speak, right? So what we need in our cell is entirely unreactive things. Do you remember back in the alcohols chapter I said an alcohol is like the first ideas of biochem? The alcohol is not a nuke. The alcohol is not a leaving group, but you could destabilize and turn it into a nuke, turn it into a leaving group. So we can't have this coexisting with a nuke. NH3 is a legit nuke. You can't have that in the cell. So in order to transport nitrogen safely, we need this urea molecule, because this nitrogen's not a nuke anymore since it's resonating with the carbonyl. And so is this. This is safe transport of 2 nitrogen. And the urea cycle is wildly important. It'll be on your MCAT. And the amino acid arginine, while it's an basic amino acid with a high side chain pKa, is part of a separate thing called the urea cycle. And we get urea by this chopping off. So just remember, arginine has something to do with it. And the remnant here is ornithine. Let's take a look at ornithine again. No, I don't want to. Screw that. I was going to say some more things, but if you look at side chains for amino acids, for example, aspartic acid, when you protonate it, it's neutral. If you look at cysteine, you protonate it, it's neutral. There are 3 amino acids that have side chains that when protonated are not neutral, they're positive. And those 3 are histidine, lysine, and arginine. So histidine has a positive side chain, lysine has a positive side chain, and arginine has a positive side chain. And it has something to do with that is what makes them the basic amino acids. There are 2 amino acids that are known as the acidic amino acids, and there's 3 amino acids known as the basic amino acids. These 2 are acidic amino acids, and this, and that are the basic amino acids. Is anybody looking at the board right now saying, okay, I see the acidic amino acids, I get it, they're the lowest pKa, but how could these be the basic amino acids? How could histidine be a basic amino acid when tyrosine has a larger pKa than histidine? Because it's not whether or not its pK is big or small, it has to do with something called the isoelectric point. Anybody ever heard of an isoelectric point? Yeah, you need to know it. So, what is the isoelectric point? The textbook gives you a formula, which is wrong. It's right in some cases and wrong as a general formula because it doesn't work for anything that has a side chain or dipeptides or polypeptides. What is the formula for isoelectric point? It says Pi, which is the isoelectric point, is the average. It says the pKa of the alpha COOH plus the pKa of the alpha NH3 divided by 2. That's what it says. That's not true. It's not right. So I'm going to show you a really simple, straightforward way of figuring out what the isoelectric point is. The isoelectric point is when the amino acid is a Zwitter ion. What is a Zwitter ion? It's sort of like a yilid. What's a yilid? The Davidic thing, right? It's sort of like a yilid. A yilid is a type of Zwitter ion, but we really don't consider a yilid a Zwitter ion. A yilid has positive and negative, but it's across a bond. This has to not be on opposite sides of a bond. There has to be like an atom in between. Because a yilid could resonate and get rid of its charge. For example, the phosphine yilid has a phosphorus with bonds and a positive charge with a carbon with a negative charge. But through resonance, the carbon and phosphorus are both neutralized, right? That you can't do that with a regular Zwitter ion. So this is the form of a Zwitter ion. Let's say the simplest amino acid, glycine. That's not the Zwitter ionic form. If I deprotonate the carboxylic acid, now it's in its Zwitter ionic form. Do you see that? And by the way, I could deprotonate it again. So I could go from here to here, and then I could go from there to there. See, I deprotonated this, then I deprotonated that. This has an overall positive charge. This has an overall neutral charge. This has an overall negative charge. The one that has an overall neutral charge but has charges in it is the Zwitter ion. So I'm going to write something over here, and then we're going to start proving it and figuring it out, right? But the acidic amino acids, the only reason they're called the acidic amino acids is because their Pi, their isoelectric point, is less than 5. Just write that down for now, and in a few moments we'll figure out how and what. And the basic amino acids, it's not Notice again, it's not the ones that have the highest pKa, because if it is, then phenol should be it and histidine should not. The basic amino acids are the ones that have an isoelectric point greater than 7. Which means what? That every other amino acid has isoelectric points between 5 and 7. And only 5 of them go out of that range. So I'm going to erase some of this table, because I need the room. There are 3 forms of glycine. If I'm talking about glycine, there's going to be 3 forms. If I fully protonate it, it's H3N, alpha carbon COOH. And if I add base, then I go that way. If I add acid, then I go this way. Everyone watching? Base go that way, acid this way. Base, I get this, H3N plus COO minus. That already is going to be a hurdle for some of us to overcome, because why did I deprotonate that? Why not deprotonate this? And why not deprotonate both? Is it random? You go to some general chemistry textbooks and they show the structure of an amino acid. They write amino acid has this general structure. And this may be true in outer space. If I shoot one amino acid into deep space, this might be what it looks like, but this is not at all correct in any kind of solution. I have that, I add some base, I get this. And if I add more base versus acid, if I add more base, then I get this. Why is that true? Do you remember the Henderson-Hasselbach equation and the table we built? Do you remember that we had, let's say, a hypothetical table like this with pH and then percent HA and percent A minus, and let's say it was for something that had a pKa of 6, just for argument's sake. Then we would say that at the pH of 6, it's 50-50. And at a pH of 5, it's actually more acidic, so you have more of the acid. It's 90% acidic and 10% basic. And at a pH of 4, it's 99-1. And you go to 7, and it goes the other way. You start with a pH equals pK, it's 50-50. And then when you raise the pH, it's more basic. So you have more of the base and less of the acid. And pH of 8, it's 99-1. So what does that mean? If the alpha carboxylic acid has a pKa between 2 and 3, and the alpha amino group has a pKa between 9 and 10, when does this group start getting deprotonated? At what pH? Let's give it an actual number, 9.5. Let's average it. Let's go with 2.5. When does this group start getting deprotonated? It's right here. See, we didn't do this so you would forget it. When does this pKa 6, an acid that has a pKa 6, when does it start losing its proton? What if I wrote 3? What would I write? About 100% and about 0%. At a 2, about 100% and about 0%. Let's go all the way this way. At 3, it's like approximately 100%, approximately 0. At 2, it's approximately 100, approximately 0. Something that has a pKa of 6 at 2 is not deprotonated. At 3 is not deprotonated. At 4, it begins. 2 units before, it begins. 2 units after, it's finished. It's already been completely wiped out, deprotonated. When does this group start getting deprotonated? At a 0.5. When does this group start getting deprotonated? At 7.5. When is this group finished fully being deprotonated? At 4.5. Did this one even start at 4.5? No, this one starts at 7.5. This is that much more acidic than when I throw base. All of these are going to get deprotonated before any of those. Which is why I got this right here. And as I add more base, suddenly this is the only thing left. The 9.5 is the only thing left to get deprotonated. That's step one for us to understand how we get the isoelectric point. Not only do students ignore what I'm about to do, but professors do also. Because you'll go to some professors and they'll say, oh, for any of these amino acids that have side chains, just take an average of all three numbers. It's insane. Because if the textbook says the isoelectric point is the average of the two pKa's, why is it an average, first of all? But if it's an average of the two pKa values, then if there's three take the average. That's crazy. I shouldn't have erased this, by the way. Let me quickly redraw this to explain why it's even an average. So, this has a pK, let's say, of 2.5, and this is around 9.5. So that means when the pH is 0, 100% of the molecules look like this. And when the pH is 0.5, it's still approximately 100% like this. And when the pH is 1.5, 10% of the molecules have this deprotonated. So 10% of the molecules look like this, and 90% still look like this. When the pH is 2.5, 50% of the molecules are like this, and 50% of the molecules are like this. Because that's the only acidic group. We ignore every other higher pKa groups. And again, when the pK is 6 and the pH is 6, 50-50. So when the pH is 2.5, it's 50-50. This is my Zwitter ion. Why Zwitter? So this comes from a German word. You know, we sometimes get German words thrown into chemistry like E and Z, eingagen, zusammen, and so on. Zwitter is a really crude word in German. It's not very nice. The word means hermaphrodite, or something that's very close to hermaphrodite. Hermaphrodite is a human being who has both sexual organs. Male and female sexual organs, born with both. So Zwitter, it's got both sexes. Positive and negative. So we want to know what the Pi, the isoelectric point, is the pH where this is at a max. We want this molecule to be maximum. Now, if I have this at a pH of 0, this is the molecule that's in my flask. If I raise the pH to 2.5, then half the molecules are this, and half the molecules are this, right? Now, there's something about this chart, by the way. It's not actually 100. It never reaches 100. In reality, at a pH of 9, it's 0.1 and 99.9. And at 10, it's 0.01 and 99.99. You see, it never reaches. Imagine there was no third form. Just as an argument here. What should we raise the pH to get as many of those as I possibly can? At 2.5, it's 50-50. At 3.5, it's 90-10. At 4.5, it's 99-1. At 5.5, it's 99.9.1. Shouldn't I go infinitely above this number? Because the more I go above this number, the more molecules I deprotonate. You guys with that? You're following? Everyone clear on that? But here's the thing. If I go too far above, this one starts deprotonating, and I get an H2N COO minus, and I don't want that to deprotonate 9.5, because then I'll get this. So how do I stop this from forming? I want to go infinitely below this number, right? The way we can play this game is you draw the fully protonated form, and then over the arrow you put the lowest pK, 2.5. Then over the next arrow, you put the next pK, 9.5. And to go that way, you want to go above that number in pH, but to go this way, you want to go below that number in pH. So what is that point? It is the average of 2.5 and 9.5, right? Now, theoretically, think hypothetically here. Will it be ever possible that it's the average of every pK? It can't be the average of pK. It would have to be the average of one and the next number somewhere would be the isoelectric point. So let's take a look at what histidine looks like. because remember, histidine was before tyrosine, right? On our list, you have it in front of you somewhere, right? You had aspartic, glutamic, and then you had histidine, right? And histidine is one of the basic amino acids. There's things past that, right? Why is histidine one of the basic? How about this? You don't even need to know what the side chains look like for the isoelectric point game. So let's say you completely forgot what the side chain looks like, but you know that it's an alpha carboxylic acid, an alpha amine, and then you know that it's some kind of side chain with a positive charge. Why? Because there's only 3 of them that had the positive charge. Histidine, lysine, and arginine. So just put a positive charge there. I don't even need to know what gets deprotonated. The pK of this, let's say, is about 2.5. The pK of this, let's say, is 9.5. By the way, it's not. It's the 1.8. I've already explained it. But let's take an average of all of them. And then the pK of this side chain is 6.0. When I deprotonate, I could deprotonate 3 times. Do you see? And when I deprotonate, I'll go above a 2.5. And when I go above a 2.5, that gets deprotonated. So it's H3N plus COO minus. And then the side chain that's still positive. Now what pK do I have? I've got a 9.5 over here, and I got a 6.0 over here. I deprotonate again. What do I go above? I go above a 6.0 number. And what do I get? I get H3N plus COO minus and a neutral side chain. Then what? Don't forget this pK right here, which is a 9.5. By the way, should this pop up on an exam, pay attention to how we do things. Don't suddenly get confused. If they ask you, what's the isoelectric point of this thing, right? And they're not going to give you these numbers. In the sentence, they're going to say, the pKa's are 2.5, 6.0, and whatever. Don't look at this. First, fully protonate it. I can't put a number here if it doesn't have an H. It's already been used. Make sure to protonate every group. 9.5, 6.0. See, I don't put numbers here because the groups have already been deprotonated. That's the last group. So when I go above 9.5, I have H2N, COO minus, and that. Do you see, what's my molecule over here? Do you realize this right here, if you add up all the charges, is histidine 2+. If I add up all the charges over here, I have histidine plus. If I add everything up over here, I've got isoelectric histidine. You've heard of isoelectronic, by the way, before, which is different than isoelectric. Isoelectric refers to having positive and negative charges and the whole thing is neutral. That's isoelectric. Isoelectronic was when you had ions that had the same number of electrons. For example, O2 minus, F minus, Ne, Na plus, Mg2 plus. All of those are isoelectronic. And the one that has the fewest protons is the largest in size, and the one that has the most protons is the smallest in size. Anybody remember this from Gen Chem? Raise your hand if you remember this. Do I average all of these pK? It doesn't make any sense, right? Here is histidine minus. So which is my isoelectric molecule? This. And in order to be here, what's the pH here? It's well above that number and also well below that number, which is the average of 6 and 9.5, which is 15.5 divided by 2, which is like 7.7 something, which is above a 7, which is what makes it a basic amino acid, right? Like on the other hand, let's try one of the other ones. This is not histidine anymore. Let's do tyrosine, because tyrosine side chain has got a larger pKa than histidine. No? Look at your table. Tyrosine side chain is a 10. Histidine side chain is a 6, right? And yet histidine's isoelectric point will be higher than tyrosine's. Why? because tyrosine, let's say, has a 2.5, a 9.5, and a neutral side chain that's a 10. Do you see? So, what is the charge of tyrosine right now? It's Y plus. Remember tyrosine, Y? Y plus, because neutral, neutral positive, Y plus. So over here, it's Y. That's already my isoelectric molecule. And which number do I go above? 2.5. And I get here, and then this is what? 9.5 and 10. And then what do I go above next? 9.5, and I'll get to this, right? Which is going to be what? This gets deprotonated, right? So that means this is now negative. So I got a negative, negative, and a positive. I have a... I lied. That's not it. Why did I deprotonate the 10 when it's a 9.5, right? That stays. That gets deprotonated, so it's H2N. And that means I have a 10 left over. And when I deprotonate the 10, now this is negative, right? Negative. So we have a Y plus, a Y minus, and a Y2 minus, if that's something we've been following. How many of you are tuned into this? Isn't this a freaking easy procedure? Like, that's what baffles me always, is when I show how to do this the right way, which is just like, oh, so just write down its charge and go -1, -1, -1, and put the numbers in order and find the one that's neutral and go above that and below that, average 2.5 plus 9.5 divided by 2. How much does that take? How much energy does that take versus getting it wrong by doing 2.5 plus 9.5 plus 10 divided by 3? Because that's the wrong thing to do. And I've had students come to me after I showed them this. They go back to the recitation professor, and the recitation professor says, not always. I'm not bashing every recitation professor. I'm just saying I've ran into these cases. And the recitation professor says, no, what you have to do is take an average of all of them. Look, there's a table that you have in your book, and that table tells you the isoelectric points. So why don't you do that? Why don't you try my method, see if you get the number right? Why don't you try other methods, see if you get the number right? It's a really simple method. What's 2.5 plus 9.5? It's 12 over 2, 6. That's a lower isoelectric point than histidine, right? By the way, not all of these are tyrosine. The isoelectric one is tyrosine. This has a net positive charge. It's tyrosinium. Tyrosinate. Inium. 8. Right? So we can make dipeptides, tripeptides, and find their isoelectric point. Wow, time flies when you're having fun. How do you draw dipeptides, tripeptides? We need to be able to get through that. I want to show you how to draw. So what is a dipeptide? If you take an amino acid, obviously you're going to need to do some kind of synthesis, but if you take an amino acid with like some group, R1, right? It could be tyrosine, phenylalanine, who knows? Some amino acid. And then another amino acid, H3N plus, COOH, with some different group R2. If you connect them through an amide bond, you'll have R1, and then you'll have what? A carbonyl right here. Do you see how that? This will be the carbonyl or carboxylic acid connecting to that, making a peptide bond. That link is called a peptide bond. We could also say it's an amide bond, but an amide bond between amino acids is named as a peptide bond. It's called a peptide bond. I drew this really funny. I never draw it this way. I'm just trying to demonstrate that this and this, there is a possibility that they react and dehydrate, and you get that plus H2O. You get a dehydration reaction. How would we do this in an orgo lab? You obviously wouldn't just put those things in the same flask, right? Why not? Well, first, why would they ever react with each other? They do acid-base, if anything. What you would need to do is, let's say, convert this into something that you could react with that. You would have to have this as, let's say, an acid chloride. And then you'd have to have this in its nucleophile form. But then that's impossible. Look what I drew. I drew that deprotonated and that protonated. And what realm of possibility is that? That's in no realm. So that means this would be deprotonated. And now who the hell's the nuke? There's bigger problems because can't this attack itself? Like if this gets deprotonated, then if this is in a beaker with millions of these, they could attach to each other. So we've got a problem because we have bifunction and bifunction, and there's no way that I could have this and this react unless I prevent all other reactions from happening. So before I ever turn this into an acid chloride, before I ever play that game, this should have already been blocked and protected. And before I ever have this deprotonated to be able to attack, this better have been blocked and protected. That's what we want to do in organic chem version of this. And then, of course, we'll deprotect and so on. The body does this also. It connects these things to tRNAs and puts them in the ribosome, and it does sort of like this protection to make sure that the only groups that you want to react are going to react. Right? You're generally following this idea, right? In the next lesson, we're going to talk about those protecting groups. I'm not done yet. I want to talk about some structural features of this. In the next lesson, we're not covering the whole chapter, just the most important parts. In the next lesson, we're going to talk about how to do the synthesis, how to protect, deprotect, so that we can use those ideas for polymer chemistry. We're going to jump right into polymer chemistry in the next lesson. But yeah, so we'll talk about protecting and deprotecting and the special reagents, but we get the idea that this is an amide bond, right? So I want to now show how, do you see, by the way, you could keep going, you could keep making amide bonds? I want to show the general structure of peptides. So let's say we had the tetrapeptide. Tetrapeptide. EGAC. What's EGAC? E, the single letter code, DE. E is glutamic acid. Glutamic acid connected to glycine, the simplest amino acid. Connected to alanine, connected to cysteine. How many peptide bonds does a tetrapeptide have? The answer is not 4. 3, right? It's like a trick question. 4 peptide has 3 peptide bonds, right? That's like how many acetal bonds does a 100 sugar polymer have? 99, because there's only one acetal bond between 2, right? So how do I draw this? You start by drawing H3N. We're going to draw it from the N terminal over to the C terminal. You start by drawing H3N. Now, I don't care if you zigzag up or down. That doesn't matter. But you're going to be consistent and continue this zigzagging up, down, up, down, up, down, up, down. We're not going to worry about the side chains or anything like that. We're worried about it's got a beat to it, this continuous nitrogen, alpha carbon, carbonyl. So just repeat that in your brain. Nitrogen, alpha, because the amino acid is H3N, alpha carbon, and then a carboxylic acid. But it's not a carboxylic acid. It connects to the next nitrogen, again, alpha carbon, carbonyl. So it goes N, alpha carbon, carbonyl, N, alpha carbon, carbonyl, N, alpha carbon, carbonyl. Now watch the flow of that. I start from this, and it doesn't matter if you go up or down. For consistency, let's go up. Just watch me do it first. I'm doing 4 of them. So I'm going to have 4 repeats of nitrogen, alpha carbon, carbonyl. Nitrogen, alpha carbon, carbonyl. And if I point so look, I pointed the corner up. The next carbonyl points down. And then after that, again, I start nitrogen, alpha carbon, carbonyl. The nitrogen will point up. The NH will point up. Alpha carbon down, carbonyl back up. If the carbonyl is up, nitrogen points down, NH down. If it's down, alpha carbon's up, carbonyl is down. Do you see it's repeating? Nitrogen, alpha carbon, carbonyl. Nitrogen, alpha carbon, carbonyl. How many amino acids do I have on the board right now? 3. Nitrogen to carbonyls and amino acid. Nitrogen to carbonyls and amino acid. Nitrogen to carbonyls and amino acid. Nitrogen, alpha carbon, carbonyl. Nitrogen, alpha carbon, carbonyl. Nitrogen, alpha carbon, carbonyl. Nitrogen, alpha carbon, and then over here I'm going to go COO minus. Because we want to draw this at physiological pH usually. Zwitter ionic form usually. Do you see that? Now since we're going from left to right with our nitrogen alpha carbon carbonyl thing, right? And we have our corner pointing up. The first R group is going to be out of the board. Do you remember the L amino acids? So the first R group I'm going to put a wedge. For the next R group, I'm not going to put anything, just because it's glycine. It just happens to be glycine. But if it was any other amino acid, I'd put a dash. Because you see the corners down? Corners up, wedge. Corners down, dash. Corners up, wedge. Corners down, dash. Glutamic acid has what? Glutamic acid, right, is like glutaric acid. It's got 3 carbons in between carbonyls. Carbonyl, 1, 2, 1 more carbon, and then COO minus. Then this has 2 hydrogen. Then over here you've got alanine. That's just a methyl group. Then over here is cysteine. It's a carbon and then an SH. What is the isoelectric point of this thing? Not so difficult at all. Don't get immediately overwhelmed. First of all, any amino acid has a COOH, has an H3N, and some of them have side chains, right? If this is participating in a peptide bond, it doesn't have a pKa anymore. If this is participating in a peptide bond, it doesn't have a pKa anymore. So the only, and now if I'm asking about isoelectric point, what was the rule? You had to protonate everything. So I'm going to protonate everything. I'm going to protonate this. I just protonated everything that could be protonated. The only alpha amine that counts is the one all the way at the end. And this I'm going to call 9.5. In Biochem 1, we're going to fix that and say there's new inductive effects when it's in a peptide, and we're going to change it. But for now, we're going to leave this original pK values. So that's the only alpha amine that counts. This is no longer a pKa. This is no longer a pKa, no longer a pKa. The only alpha carboxylic acid that counts is the C-terminal, all the way over here. Does everyone follow? Because all the other carboxylic acids have been peptide bonded. They're not an acid anymore. So this one we're going to say is 2.5. And then, of course, any side chain counts, right? So the side chain of a glutamic acid is what? 4.1. The side chain of a glycine is nothing. The side chain of an alanine is nothing because it's not even on your list. And the side chain of a cysteine is an 8.0. I wanted to show you how to do it there before I do that up here. Here's the game that we play up here. I really should have put something like a histidine over there, but let's say I did have glutamic acid. Let's say I had glutamic acid, glycine, histidine, and then cysteine, right? You don't have to draw the whole thing out to figure out the isoelectric point of this thing. You would do this. Over here, all the way to the right, you'd write COOH. All the way to the left, you'd write H3N+. Do you see why? The N terminal and the C terminal. Underneath, I'm going to write pKa's, and above them, I'm going to put charges, if there's any kind of charges. So what's the point? Look, up here, I put a positive, because I know that has a positive charge, and that is a 9.5. Glutamic acid is a 4.0, and above there, I'm going to write N for neutral. Remember the side chain is neutral? It's only 3 amino acids that have side chains that have any kind of pK that you have to, that have positive charges when protonated, is what I meant to say. Histidine, lysine, and arginine. So look, glycine has nothing. Histidine is a 6.0, and this is one that has a positive side chain. Cysteine is an 8.0, and it has a neutral side chain. You with that? And then this alpha carboxylic acid is a 2.5, and it has a neutral. So here's how I deal with this. I would write PEP for peptide and add up all the charges. 1, 2 positive charges. Do you see that? So it's a peptide 2+, which means if I deprotonate, it becomes peptide plus. Deprotonate again, it becomes peptide. Deprotonate again, it becomes peptide minus. How far can we deprotonate? Who cares? I just need to go till it's neutral and then one more so I can put the numbers. The lowest number is a 2.5. The next lowest number is a 4.0. The next lowest number is a 6.0. The isoelectric point of that thing is 5. You see how it's 5? Relatively easy, right? What's the isoelectric point of this? And it would completely screw you up, by the way, if you had a negative over there. Because do you see, look what's wrong. If I had a negative, but I still left my 2.5 number there, and if I had a negative right here, but I left my 4.1 number, I'm starting with the wrong charge. It doesn't have a negative charge when it's fully protonated and these numbers are used. This is, and look, on an exam I wouldn't write PEP either. I just write X. Why spend the extra time for the extra 3 letters, right? 2 letters. So this is X what? Positive, neutral, it's just positive, right? This is X plus. Deprotonate once, it's X. Deprotonate twice, it's X minus. How many times can you deprotonate? As many as you have pKs. This could be deprotonated 4 times. But to find the isoelectric point, I just need to deprotonate twice because I'm starting with just 1 overall positive charge. What's the lowest pK? 2.5. What's the next lowest pK? 4.1. I'm giving you tetrapeptides. It's easy to look over the molecule and figure this out. When we get to, let's say, some tougher questions, and not tougher, just like more involved, you're going to have like an undecapeptide. So you'll have like 11 amino acids. Now what happens in that case is you're worried and you're like looking at the molecule, and by accident you could count the same pK twice. So what I would do when I'm writing this down is when I write a 2.5, cross it out in the main molecule so I don't accidentally write the same number twice. You follow the idea? So this is easy, 2.5 plus 4.1 divided by 2, and that's the isoelectric point over here. There is this, we're done in 5 minutes. There is this way of figuring out the actual isoelectric point. It's very interesting. And here's some orgo. Ready for orgo? There's this way of figuring out an isoelectric point by this technique called paper electrophoresis. And what that is you take, you have this tray where you put a buffer in there, and you could put whatever pH for the buffer you want, right? And there is a location where you put a paper, and you stain it with an amino acid 2, 3, however many you want. Like, say, pipette at some amount of amino acid over there. And you have an electrode and another electrode, and these electrodes are connected to a battery. So if this is connected to one terminal of the battery, it'll be negative. If this is connected to another terminal of the battery, it'd be positive. And then, of course, if the amino acid is positive, it would repel the positive, attract the negative, and move this way. If it's negative, it would go that way. If it's neutral, it wouldn't move at all. So let's say I had 3 amino acids. Let's call this A, let's call this B, let's call this C. And let's say A had an isoelectric point of 4, and B had an isoelectric point of 5, and C had an isoelectric point of 6. If I put this all into a solution, like from a side view, by the way, from a side view it looks like this. It's this tray that in here there's an electrode, in here there's an electrode, over here there's a paper, and this whole thing is submerged in buffer. And the buffer, right, I'm looking at the side view. If I look at it from the top, then it looks like that. So if it's all submerged in buffer, let's say the buffer pH happens to be 5. What does that mean? What would this be? Well, if something has an isoelectric point of 4, then what would it be if a buffer pH is 5? Sometimes it's hard to wrap your head around that, but let's take a look at, for example, glycine, right? Glycine. Glycine was H3NCO with a 2.5 and a 9.5. And then if you went above 2.5, it would be an H3N plus COO minus. And then if you went above 9.5, it would be H2N COO minus. Here, glycine would be positively charged. Here, glycine would be neutral. Here, glycine would be negatively charged. At its isoelectric point, it's neutral. And at pH lower than that, it's positive. And at pH higher than that, it's negative. The way that I remember this, I know that it's going to be tough to wrap your head around this. You'll get it when I say it, but then when it's time for you to think about it, I always start this way. What if this was neutral? At which pH would this have to be neutral? It would have to be neutral at a pH of 4, right? But if the pH is 5, that means I raised the pH, I made it more basic. Now it's negative. More basic, negative. This would be neutral at if its isoelectric point is 5, it would be neutral at a buffer 5. It's in a buffer 5, it's neutral. Again, what pH would this be neutral? This would be neutral at a pH of 6. So let's say I start at pH of 6, it's neutral. And then I drop the pH to 5. If I drop the pH to 5, I add acid. Acidic, positive. Does that make sense as the way to wrap your head around it? I think it's a nice systematic way of wrapping your head around it. Otherwise you'd have to memorize. When the pH is greater, then do this. When the pH is less, then do that. And that always tripped me up. Always in a moment of nervousness, I was like, shit, was it bigger or smaller? But if you start out where it's neutral, to go at a higher pH, you're adding base, negative. To go at a lower pH, you're adding acid, positive. What would that mean? That would mean A, at this buffer pH, would be negative, and it would move this way. B wouldn't move at all, and C would move this way, right? So you put them on paper. On the paper, you let this run, they spread out, and at some point you take off the paper, you look, and you see absolutely nothing. Because amino acids don't absorb any visible light. And the reason they don't absorb visible light is because there's very few that have aromatic. None of them have charges in that aromatic. They're not transition metals with ligands. They're not conjugated systems. They're not aromatic charges. So these things don't have any. So basically, you take the paper out and you don't see anything. Also, if you end up doing homework questions on this, a lot of people get kind of really messed up when this is called the cathode and this is called the anode. You start questioning your existence there, like, did the book make a mistake? No, the book doesn't make a mistake. Nobody said the cathode was the positive electrode. The only thing that was true about voltaic cells, or galvanic cells, the only thing was true in this case, was that when you had a salt bridge, cations migrated to the cathode, and anions migrated to the anode. And that's always going to be true no matter what system you use. And sometimes the cathode is positive, and sometimes the cathode is negative. But here, C is what? A cation. And it's going this way, so this is the cathode. Right? So here's the issue. When these move, because amino acids are invisible, you're not going to see anything. So what you do is you take the paper out and you spray it with something called Ninhydrin. And Ninhydrin is the staining solution that breaks down amino acids, and you see a purple-colored stain for 19 out of the 20 amino acids. One of them, called proline, gives an orange color. And this staining solution is really important because if I had Ninhydrin right now, and let's say somebody broke into my office and I had Ninhydrin, I would spray it on the doorknob, and then eventually I would get purple-colored fingerprints because anything you touch, you leave your proteins from your fingers. So I would take a piece of tape, and then I'd have their fingerprints right on the Scotch tape, kind of a thing. If you go to John Jay College of Criminal Justice, they all know about Ninhydrin. I think you should know about Ninhydrin. Here's the structure of Ninhydrin. I mean, I drew this before, but that just means you had to take all the notes and remember. This is when we were talking about hydrates. I talked about chloral hydrate. Remember I said rank these in order of who wants to make the most hydrates, and aldehydes made more hydrates depending on withdrawal effects. So I said there's something called chloral hydrate, where if I put 3 chlorines over here, it's so withdrawn that this doesn't even exist as such. It exists just as a hydrate. Same thing here. There's supposed to be a carbonyl over here, but it's got a carbonyl and a carbonyl, so it's so withdrawn that it's a hydrate. And this thing reacts with an amino acid. This thing reacts with an amino acid and gives you a derivative that's usually purple in color, except for the proline case, it's an orange color. And it's very clearly written, the entire reaction. I guess I'm going to show it. H3N, and then you have some kind of R group, and then you have COO minus. And when this reacts, it reacts with 2 of these ninhydrin. And the result is this purple-colored anion Does it make sense at all that this would give you some sort of color? Do you see it's not only like aromatic conjugated system, but it's also got a charge, and this thing is resonating like this, down here and through here and into this side? That makes sense. Where did the nitrogen come from? They came from the amino acid, right? So, but here's the orgo, because you guys have been missing this. So nitrogen has been ripped off of this compound, right? Do you see this bond had to break somehow in order to get the nitrogen in there? Not only that, the reaction produces CO2, and so that means somehow decarboxylated. So that got broken off. And it produces an R aldehyde. So it's an R carbon, and that carbon is an aldehyde. From there to there. Find this in your book, because right next to the picture of the paper electrophoresis in this chapter, it shows this mechanism, this reaction. And for homework, I want the mechanism. And many people before you have given me the mechanism. And I'm not talking about 10 years ago. I'm talking about 2 semesters ago, the last time I taught this course. People have done this because this tests, look, there's a deamination, there's a decarboxylation. Do you see what I'm saying? There's the do you see an imine bond right here? There's imine bond formation. There's hydrate removal. There's like literally 10 different mechanisms cooked into one mechanism. We have a final coming up. You need to practice some mechanistic ideas. Get it right, get it wrong, who cares? I'll give you one hint. I've seen students start this mechanism off, and the first thing they've done is a decarboxylation. Now, here's the only reason why that's bizarre. Because amino acids exist. If you're going to just decarboxylate it, that means it wouldn't exist. That all amino acids would just decarboxylate themselves. You can't just decarboxylate it. You better like link it to something and then give it a reason to decarboxylate through the process. What's a reason to decarboxylate? When you have electrons that can move in a beta position. I don't see any beta position here. But maybe if this nitrogen links up over here and makes a double bond over here, then a carboxylate alpha beta to a double bond, that would be a reason. You hear what I'm saying? We'll finish up some ideas from this chapter and then go right into polymers in our next lesson. Thank you for your participation. Please hit like and subscribe.